Okay, welcome everybody to the latest talk in our series of talks from Fungisphere, which is a multidisciplinary research node in the Sydney Infectious Disease Institute at the University of Sydney, interested in all things mycology. We're trying to keep it broad, and so including human, animal, and plant environmental health. We've even just had a pre-discussion about The Last of Us and the current TV show that's ongoing and causing lots of excitement for people who are interested in fungal pathogens and zombies. Not what we're talking about today. I'm going to hand over to Julie Djordjevic to introduce today's speaker and to just give us a little snap of what is upcoming over the next hour. So thank you, Julie.
Thank you, Justin. I'm sure all of you know Desmarini's, but just a little bit of background. So Desmarini is doing a PhD in my laboratory, and she's a recipient of one of these PhD partnership program fellowships, which is designed to build capacity in the New South Wales health system. And today she's going to be talking to us about a special class of kinase called an inositol polyphosphate kinase, and how these kinases might be good or potential targets for antifungal drug development. So with that introduction, I'll let Desmarini tell you more about it. So thanks, Des. If you'd like to start sharing your screen.
Julie, for the introduction. And I also like to thank the organizer, Justin, for the opportunity to speak today. So I am the only member in my family who works in science. So when I tell my friends and extended family circle that I research on treating fungal infection, they often associate it with toenail infection. Not that toenail infection is not important, or this audience might know that toenail infection is really difficult to treat. But fungal infection is more than just those superficial infections. It can also invade our internal organs, such as brain and lungs, to cause potentially fatal invasive fungal infections. And they affect the ever-increasing immunocompromised individuals, killing 1.5 million people annually. It is an important yet often neglected issue that the WHO recently released a fungal priority pathogens list. This list was compiled by a group of experts, including Justin in the audience. And I have listed here a few of them, and Cryptococcus neoformans and Candida albicans, which I'm going to talk about today, fall within this critical priority group, which is the highest category based on funding support and difficulty to treat. And humans are not the only ones affected by these. Fungal diseases also threatens the animal and plant kingdoms. For example, the white nose syndrome, that causes mass mortalities in bats, and the chytridiomycosis that is responsible for the extinction of around 90 different amphibian species. And fungal pathogens also destroy plants, which threatens food security. So why is treatment of fungal disease difficult? Well, just like human, animals, and plants, fungi are also eukaryotes and therefore share a lot of common features. And this makes developing a safe and effective antifungal drug challenging. Currently, there are limited classes of antifungal drugs and only 4 classes of antifungal drugs to treat human systemic infections. And most of them target either the cell wall or the ergosterol, which is the most abundant sterol in the fungal cell membrane. And this treatment issue has been made worse by the emerging antifungal resistant strains. I'm sure this audience will have heard of the multi-drug resistant Candida auris. So there is an urgent need to develop new antifungal drugs, and my project is to address this by developing a new drug class. So for drug discovery, typically there are 2 different approaches. The first one is by library screening, where hundreds and thousands of compounds are screened by various methods. And once you get a compound which is antifungal, or what we call a hit, you then characterize it, look for the mechanistic insights, etc. The advantage of this method is that you get a compound which is already antifungal, but the downside is that you don't have the mechanistic insights, so you know very little regarding its potential safety or off-target effects when used as human therapeutics. And the few hits that you get, if you're lucky, can sometimes also be false positives. The second approach is a more targeted approach, where years of basic research is required to identify a potentially druggable target. And this is the approach that our group is taking. So we have a target, which I'm going to talk about very soon. And we know the mechanism of it, and we work with medicinal chemists to develop compounds to inhibit our target. And of course, there are also more work with this approach. We need to make sure that the compounds do get into fungal cells. And once it gets into the fungal cells, they will need to have an antifungal effect. So our target is this inositol polyphosphate kinase, or IPK pathway. This pathway is an essential pathway that regulates many cellular processes, and it is conserved in both human and fungi. So our lab uses Cryptococcus neoformans to study this pathway in pathogenic fungi. But before I go further into the pathway, I'd like to talk a little bit more about these small molecules. So this is an inositol ring, and it has 6 carbons, and inositol polyphosphates differ depending on how many and where the phosphates are. For example, there are phosphates on positions 1, 3, 4, 5, and 6 on this inositol, and there are 5 phosphates altogether. So this IP species is called inositol 1, 3, 4, 5, 6 phosphate, or IP5 in short. In some cases, double phosphorylation could occur. This then becomes 5-ppIP4, where pp refers to the double phosphorylation, 5 refers to the carbon position where double phosphorylation occurs, and IP4 refers to the inositol and the remaining 4 phosphates on this inositol. So different phosphorylation positions result in a different inositol polyphosphate species, which are generated by different inositol polyphosphate kinases. So the kinase that phosphorylates IP5 is called IP5K to generate IP6, and then the kinase that phosphorylates IP6 is called IP6K to generate IP7, and so on. So all these different IPs and IPKs have different metabolic role, and the kinase can have catalytic functions, and which is to generate this different inositol polyphosphate, and also non-catalytic functions. For example, they can act as adapter protein and stabilizes protein complexes, and the inositol polyphosphates themselves are biological effectors, and they can also have protein complex stabilizing role. So the take-home message from this slide is that there are many different IP species generated by different IPKs, and all the IPs and IPKs have important metabolic functions. But for today's talk, we are focusing on the catalytic functions of IPK, which is to generate the different inositol phosphate species. So back to the pathway now in Cryptococcus neoformans. So we have characterized this pathway using gene deletion analysis and metabolic profiling. So the metabolic profiling is where we label the cells with radioactive inositol, extract the cell lysates, and then resolve the inositol polyphosphate in the cell lysate using high-performance liquid chromatography. It's okay if you don't know HPLC, but the readout for this method is something that looks like this, where the different IP3, IP4, IP5, 6, 7 standards will be used in the assay. And then by comparing the standards and the traces that you get from the cell lysates, you'll be able to quantify the different species in inositol phosphates in the cell lysates. So using this method in combination with gene deletion analysis, our lab has fully characterized this pathway in Cryptococcus neoformans. So this pathway is linear, consisting of a series of inositol polyphosphate kinases that sequentially phosphorylate inositol to generate different IP species. And for today's talk, I'm going to focus on the early component of this pathway, the IP34K. So to study the role of IP34K, we deleted the IP34K encoding gene, which is called ARCH1 in Cryptococcus neoformans. Okay. No, sorry. And this graph here actually shows the inositol polyphosphate traces with the HPLC method. So this black line here shows the IP traces of the wild-type Cryptococcus neoformans. You can see the IP3, 4, 5, 6, 7, 8 separated very clearly. And the red line here is the inositol polyphosphate traces of the ARCH1 or IP34K deletion mutant in Cryptococcus neoformans. So IP34K in Cryptococcus neoformans is called ARCH1, and I'm going to use these two terms interchangeably, but they refer to the same IP34K protein. So deletion of ARCH1 results in accumulation of IP3 and the loss of other inositol polyphosphate species, confirming that ARCH1 is the IP34K or the early component of this IPK pathway in Cryptococcus neoformans. We then observed the resulting phenotypes from deleting ARCH1 using a spot dilution assay. So cells are grown in a media supplemented with various chemicals or grown at different temperatures. And when the conditions are optimal, such as this panel here, where cells are grown in rich media and incubated at the optimal temperature of 30 degrees, cells grow healthily down to the most diluted 10 cells per drop. But at the elevated temperature of 37 degrees, which is the human body temperature, the ARCH1 deletion mutant has growth defect. This mutant is also sensitive to the cellular-perturbing agent Congo red and has trouble metabolizing alternative carbon sources other than glucose, such as glycerol and lactate. And most importantly, ARCH1 deletion mutant cannot establish an infection in mouse infection model, as shown in this survival curve, where up to 60 days post-infection, the ARCH1 infected mice have 100% survival rate compared to the wild-type infected mice in the blue line here. The mice have median survival of 15 days. So this loss of virulence is really encouraging from a drug targeting perspective. And the reconstitution of ARCH1 restores all the defective phenotypes, including virulence, as you can see in this green line here, where the reconstituted strain infected mice have 16.5 median survival days. And I mentioned previously, very briefly, that kinases can have catalytic and non-catalytic functions. And all inositol polyphosphate kinases have this conserved PDKG motif. So PDKG are the amino acid residues known to bind to substrate to give inositol polyphosphate kinases their catalytic functions. So to investigate whether the phenotypes that we're seeing, or in other words, whether fungal ARCH1 functions are due to its kinase activity, we created a kinase dead mutant using site-directed mutagenesis. So I replaced the aspartic acid D and the lysine K, these two residues, with alanine A. And the resulting kinase dead ARCH1 mutant has similar phenotypes to the full deletion mutant, where it is sensitive to high temperature and cellular perturbing agents, and also has difficulty metabolizing alternative carbon sources. So this suggests that kinase activity is the main contributor to the role of ARCH1, and that ARCH1 has no non-catalytic functions. So overall, our data shows that IP34K, or ARCH1 in Cryptococcus neoformans, regulates many essential cellular processes, such as secretion, metabolism, phosphate acquisition, cell integrity, and the ability to grow at human body temperature. And this all made ARCH1 a good candidate antifungal drug target. Another advantage of targeting ARCH1 for antifungal drug development is its pathway redundancy in humans. As I mentioned before, and as you can see in this graph, in this scheme, that the IPK pathway in fungi is very simple and linear. But the IPK pathway in human is much more complex, where the products of our target, IP4 and IP5, they can be generated through alternative routes. There is also low sequence homology between the fungal and the human enzymes to only 21% identity. So this indicates that there are enough differences between the fungal and the human enzymes to selectively target the fungal enzyme. A good example of proof of concept that selectivity could be achieved is the olorifim, which is the new antifungal drug in the orotomide class that is currently in phase 2 clinical trial. So olorifim interrupts pyrimidine synthesis, which is a pathway conserved in both human and fungi, just like IPK pathway. And olorifim inhibits the enzyme dihydroorotate dehydrogenase with over 2,000-fold more potency against fungal enzyme compared to the human enzyme. So our strategy is to target the early component of this conserved essential pathway, which has a role in many cellular processes, as opposed to the current antifungal drugs, which only have one target, which is the cell wall or the ergosterol in the cell membrane. And to help drug discovery, although not essential, having structural information can only help. So we tried to crystallize Cryptococcus arch1 to obtain protein structural information, but so far we have no luck and this process is ongoing. But AlphaFold, which is an artificial intelligence prediction program, it has recently released the predicted structure of over 200 million proteins, including Cryptococcus arch1. So using this information, we were able to create this surface plot for Cryptococcus arch1 and predict the arch1 ATP binding pocket and IP binding site. So we propose to develop arch1 inhibitors that bind to this ATP binding pocket, since there are many approved drugs that target this ATP binding pocket, especially for protein kinase inhibitors. So we have a couple of different approaches. The first approach is by fragment-based drug design, or FBDD. This to develop a new molecular entity, and this is in collaboration with Sydney Analytical. So with this approach, we start by looking for small fragments which are not drug-like at all, that with the right fit into this chemical space or the small pocket of chemical space of this binding site. We then either grow the fragment or link the various small fragments that fit into this chemical space into more drug-like compounds. So the advantage of this approach is that there is a higher chance to develop a potent and specific inhibitor, but the downside is that because we're starting from scratch, it might take a lot longer, depending on luck. So far, we have identified 2 potential core structures that we could develop, but this approach is still at its early stage, so I won't elaborate any more on that. The second approach is where I'll focus on today, which is our medicinal chemistry approach. So we have 2 collaborations, one with Monash University and another one is with University of North Carolina. So as part of the development, there is groundwork that needs to be done. So first, we purified the recombinant IP34K enzymes from human and from 2 fungal pathogens. And this is a protein gel showing the pure enzyme from Cryptococcus neoformans and from Candida albicans. And the human enzyme is about 60% pure, but we can work with this purity for our next step, which is the enzyme assay to screen for potential inhibitors. So IP34K is a kinase, so it hydrolyzes ATP to phosphorylate IP3 to generate IP4. So I use a kit where ATP is converted to luminescence. I then tested the compound dose response and observed the ATP remaining as a function of luminescence to generate this curve. And to assess potency, we use what we call an IC50 value, which is the concentration at which ATP amount is reduced by half. So what we want to see is this curve sitting on the left side of the graph, indicating low IC50 values. So we want to use minimum amount of drug to get maximum antifungal effect. So now that the groundwork is done, we can start our inhibitor development. And I'll start with our Monash collaboration with Phil and Daniel. So we use a compound called TNP as a starting point. So TNP is a mammalian IPK inhibitor. It was originally discovered as an IP3K inhibitor, but later found to be a more potent IP6K inhibitor and shown to have anti-obesity and anti-diabetic effects in high-fat diet mouse model. However, as a probe and as a drug, TNP has low solubility, which impacts its potency, and it also has off-target effects in that it inhibits human cytochrome P450 enzymes. But recently, medicinal chemistry efforts to improve TNP properties have shown success, where TNP could be modified to have improved selectivity for human IP6K1 over other IP6K isoforms. Another modification was able to reduce off-target effects by reducing inhibition of human cytochrome enzyme. So these studies were done on human or mammalian system, but whether TNP inhibits IPK of a fungal pathogen such as Cryptococcus neoformans or Candida albicans has not been investigated. So this is the basis of our collaboration with the Monash team. These studies, despite being done on mammalian system, they provide proof of concept that if TNP does inhibit fungal IPK, it is possible to improve properties of TNP as an antifungal drug by improving its selectivity for fungal enzyme, which is important for our antifungal drug development. So TNP has a purine core structure with 2 benzyl rings at positions 2 and 6, and the Monash team made modifications on these 2 positions to generate different TNP analogs. I then assessed the potency of these analogs using the luminescence assay, and this is a graph summarizing the IC50 values of some of the analogs against Cryptococcus IP34K. We found that TNP, a mammalian IPK inhibitor, also inhibited the crypto IP34K. And some of the analogs inhibited cryptoenzyme with similar IC50 values. The Monash team synthesized 45 different analogs, but none of them inhibited the Candida enzyme. But since they are active against Cryptococcus, I tested these compounds against the human enzyme to see if selectivity has improved. And we were able to see improvement in selectivity for Cryptococcus enzyme. So this is a graph showing the IC50 ratio of human versus the Cryptococcus enzyme. So the ratio, if the ratio is below 1, it indicates that the analog is human selective, and above 1 indicates fungal selective. So we started with TNP, which is human selective, and we were able to improve the selectivity for the fungal enzyme, as represented by these blue dots here with IC50 ratio some well above 1. But as I mentioned previously, the potency did not greatly improve. Our most potent compound has IC50 around 10 micromolar compared to TNP of around 20 micromolar. So we now have wrapped this work up in the form of a publication, and you are very welcome to read this paper for more details. But for today, I'd like to draw your focus on the fact that there are several analogs with modest improvement in potency and selectivity, which is encouraging. So we decided to dig a little further. So the Monash team made more structural modifications on these 2 positions, but this time the modifications resulted in analogs with structures significantly different to our parent compound TNP. So I hope you can appreciate the need to protect our intellectual property, so I'm not showing the full structure. I'm just going to call these new analogs as DT analogs, following the initial of Daniel, who made these new compounds. So I used the luminescence assay again to assess potency, and this blue line here is the IC50 curve of a TNP analog with IC50 around 20 micromolar. And 5 out of the 11 DT analogs synthesized have their curves significantly shifted to the left, which is what we want, as it means that they have lower IC50 values. And our best analogs, 318 and 155, have sub-micromolar IC50 values, which is about a 60-fold improvement compared to the earlier TNP and its analog series. And even better, these 2 analogs also inhibited the Candida enzyme, which we did not see in our earlier TNP analog series. This is really encouraging, as it means that we are starting to see pan-fungal activity of our analogs. I also tested the antifungal activity of these new DT analogs using a standard microbroth dilution assay, which is done in a 96 well plate. And this is one example against Cryptococcus neoformans. So this column here has a clinically used antifungal amphotericin B alone, starting from 2 micrograms per mil, 2-fold serially diluted down. And this first clear well indicating no fungal growth is the minimal inhibitory concentration, or MIC, of amphotericin B. This row down here has the analog 318 alone, starting from 100 micrograms per mil, 2-fold serially diluted across. There is no growth inhibition of the fungal cells, as indicated by this white colored cells in each well up to the tested concentration of 100 micrograms per mil. But the combination of the 2 drugs in this middle panel here results in a 4-fold reduction of MIC for amphotericin B and reduction of MIC of 318 down to about 3 micrograms per mil. I use the same assay to test the antifungal activity of the remaining DT analogs, and this is the summary. So the MIC for DT analogs are represented by solid bars, for amphotericin B by shaded bars, and against crypto in black colors, and against Candida albicans in pink. So I'd like to draw attention to this left side of this graph first. So as single agents, or when used as drug alone, none of the DT analogs inhibited the fungal growth. But then in combination with amphotericin B on the right side of this graph, you can see an overall reduction, except for 317. There is an overall reduction of MIC for both the DT analogs and also for the amphotericin B. So this is where our progress is so far with our Monash collaboration. So in the remaining few minutes, I'd like to just talk briefly about our collaboration with University of North Carolina with Professor Xiaodong Wang. Again, for potential intellectual property purpose, there will be no structures. So we received 11 compounds so far, and I used the same luminescence assay to obtain the IC50 values against both fungal pathogens. And 10 out of the 11 compounds are potent inhibitors, with some possessing low nanomolar IC50 values and some in the low micromolar IC50 against IP34K from both fungal pathogens. And this is between a 10 to 700-fold more potent compared to TNP, which inhibits only the Cryptococcus enzyme with IC50 around 20 micromolar. However, since these compounds were synthesized as human IPK inhibitors, unsurprisingly, all of these also inhibit the human IP34K. So this is the IC50 ratio of human versus fungi, and I have included TNP again for comparison purpose. So again, the IC50 ratio below 1 indicates that the compounds are human selective, and above 1 indicates that they are fungal selective. So TNP has IC50 ratio of around 0.3, which means that it is slightly human selective. But most of Xiaodong's compounds have even lower IC50 ratio compared to TNP, indicating that they're even more human selective. For example, for compound 8 here, the IC50 against the human enzyme is about 40 to 50-fold lower than the IC50 against the fungal enzyme. This is of course the complete opposite of what we want to see, but at the same time, this is not necessarily bad news, as it emphasizes that there are enough differences between the human and the fungal enzymes to potentially create IPK inhibitors which are fungal selective for our antifungal drug development. So I also tested these compounds for antifungal activity using the microbirth dilution assay in 96-well plates, and this is the graph summarizing the assay. And again, on the left panel here, you can see when used as drug alone, the compounds from University of North Carolina did not inhibit any fungal growth up to the tested concentration of 100 micrograms per mil. But in combination with amphotericin B, there is again an overall reduction of MIC for both Xiaodong's compounds and also for amphotericin B. And only 2 compounds showed these promising results. The remaining did not have any antifungal activity, either when used as single drug or in combination with amphotericin B. So this is where I'll wrap up my talk. I've shown you that fungal IP34K is a good candidate for novel antifungal drug development, as it regulates many essential processes in fungi, and it is important for virulence. And it is also different to the targets of currently marketed antifungals, which only have one target. There's also a functional redundancy in the human pathway. And in our efforts to develop IP34K inhibitors, we now have several promising leads. Our medicinal chemistry efforts on TNP in collaboration with Monash has resulted in a new family of compounds, which are potent fungal IPK inhibitors, are antifungal when used in combination with amphotericin B. We also have a couple of compounds with different structures to TNP in collaboration with Xiaodong's team. So our next step would be to do some inositol polyphosphate profiling on fungal cells treated with these compounds to confirm the in vivo targeting of these promising candidates. And lastly, I'd like to just acknowledge the financial and technical support and the intellectual contributions towards this project. This project is funded by Australian NHMRC grants, the Sydney Infectious Diseases SID Grant Initiative, and the Sydney Drug Discovery Institute SID grants. And also received an industry partnership scholarship from New South Wales Health and University of Sydney. And this project will not be possible without the involvement and support from these research scientists from different institutes. And I thank you for your time.
Well, thank you very much, Desmarini, for a really clear talk on a subject that's quite difficult to explain. So well done. I just thought I might just start off by asking a question. I just wanted to, I mean, you focus a lot on, obviously you use Cryptococcus to characterize the inositol polyphosphate kinase pathway. And, you know, we've established that, you know, the IP3 kinase is really important in virulence. But, I mean, do you want to just tell the audience a little bit about what we know about IP3 kinase from Candida and its contribution in Candida?
Okay, so in Candida, the study so far suggests that it is also an essential gene, as the attempt to delete both alleles of the IP3/4K encoding gene in Candida was unsuccessful. But a group, I believe in China, was successful in creating like a knockdown mutant where they deleted only one copy of the gene. And this mutant has I believe it's hyperfilamentation. And there's changes in the phenotypes of Candida for sure. Okay. But in Candida, I don't think that there is metabolic profiling performed on the Candida species.
Right, okay, but it sounds like it's even more important in Candida than it is in Cryptococcus, which is nice from a drug targeting point of view.
Yeah.
Yeah. Does the audience have any questions at all?
We have
two. Justin.
Tanya first, then me.
Oh, sorry, Tanya, I didn't see your hand up.
That's all right. I'm sort of in the dark here. Des, that was a really terrific talk. Thank you very much. As Julie said, it was extremely clear. I suppose the questions that arise from your observations about synergy with amphotericin, etc., lead me to ask you two things. The first of them is the ability of these compounds to penetrate the fungal cell, and whether that's relevant to the apparent lack of activity. And the assumption may be that amphotericin might kind of open up the cell, if you like, to entry of the material. And the second one relates to, is it a solubility issue?
Okay, I think I'll answer the second question first, which is that it is not a solubility issue, as this compound dissolves pretty easily in the media that was used to do this susceptibility or microbubble dilution assay. Mm-hmm. And your first question, I think you already have the answer there. We also believe that the amphotericin B, okay, so the compound, we believe that the compound has trouble penetrating the fungal cells, and that the addition of amphotericin B actually does act as membrane solubilizing agent, as opposed to aid the penetration of our compounds into the fungal cells, and that's why we're seeing the effect.
So that just leads to a follow-up blue sky question, which is, would it be possible to use nanotechnology or some form of nanoparticle to help it get into the cell as a future strategy?
We do have that in the plan.
Oh, okay.
Yeah, we have a collaboration with, can I say who, Julie?
Yeah.
No, okay, so with Stuart Fraser and Marcella Bilek, they have a type of plasma generated nanoparticles, and it's tagged with a
you can't say Des, you can't say what it's tagged with, okay. But you can just say that it, yeah.
So yeah, but that's next in the plan to test to pretty much do the microbubble dilution assay in the same manner, but instead of using amphotericin B, we're going to use the nanoparticles to see the effect.
Thank you.
Thanks, Des. I really enjoyed the talk as well. It's very complicated, and you made it seem pretty straightforward. So thanks so much.